So the idea of this panel is we're going to talk about research in collagen 6 and maybe collagen 6 related disorders. And the way we're going to do that is by having a discussion. So we have lots of distinguished researchers that do really hands-on bench level, research on collagen 6. And we'll go around and have everyone introduce themselves and their research. And then we're going to have, after each one, questions and then an ongoing discussion. It's important that you are not shy about it. So if you have questions, sometimes people who spend their life at the bench, they kind of go on a level that is hard to follow. And so I'm here to help translate, and everyone is going to do their best to bring the research down to a level that we can all understand. But don't be shy to ask questions. If anything that kind of feels weird or not clear to you, just ask, and that increases the fun. Sometimes I'm going to ask for you if I feel that needs to be done. So my name is Carsten Bonnemann. I'm a pediatric neurologist and physician scientist at the NIH, National Institutes of Health, which is 2 hours by train, 4 hours by car, south of here. And that's in Bethesda, close to Washington. And so my team works in a campus, the NIH campus, at an institute called National Institutes of Neurological Disorders and Stroke. Before that, I was across town here at CHOP, Children's Hospital of Philadelphia, and I still see patients there. And so there's a nice ongoing connection between the two. So our research at NIH is really collagen 6 focused, and I will give you just a couple of sentences of what we are doing, and really the history of collagen 6 patient engagement and research is really starting here in Philadelphia at CHOP. There's some people of the first hour still here. I think our first collagen 6 patient meeting at CHOP was more than 20 years ago. And yes. You all remember that. And so it's been a long time, and a lot of things have happened. I will start by just briefly outlining where I see some of the kind of burning questions that we need to answer for collagen 6. And many of you know that collagen 6 and collagen 12, for that matter, are disorders of the extracellular matrix of the muscle. And in collagen 6 and also for collagen 12, we not only have more than one gene. We have 3 collagen 6 genes, alpha 1, alpha 2, and alpha 3. But we also have 2 genetic mechanisms that we need to think about. One is called recessive, where there is disease-causing changes in the gene on 2 copies, one from the dad and one from the mom. So that's a recessive situation where there is too little functional collagen. And that's true for COL12 and COL6. And the other mechanism that we have to deal with, which is actually arguably more common, is the so-called dominant mechanism, where we have a disease-causing variant on only one of the 2 copies of the gene. So you remember, everyone inherits 1 copy from mom and 1 copy from dad. And in the recessive, you have a variant that causes disease on both of these copies, but the carrier mom and the carrier dad are unaffected. In the dominant situation, you have a disease-causing variant in only 1 copy of it. And then if it's inherited, the parent may already have some signs of the disease as well, and the child has some signs as well. Or it is a new change in the child, a de novo variant that occurs during passing the genes on to the child. So that's a de novo, and that's a more common form of collagen 6. And the reason why that is happening is because the dominant copy of the gene makes a copy that is interfering with the normal copy. We call that dominant negative or toxic gain. So having that variant with a dominant change on it will interfere with the normal copy of collagen 6 so that there is little functional collagen 6 left. If you only had 1 copy of collagen 6, or collagen 12 for that matter, that is not interfered with, you don't have disease. You only need 50% of the collagen. But it's the dominant interference that causes disease. So 2 mechanisms. Either you have way too little because you have problems in both genes, or you have a dominant mechanism that interferes with the normal copy. Keep that in mind when we talk about some of the approaches here. So that's one challenge. sorry. The second challenge is that the disease-causing problem is in the extracellular matrix. So that causes problems with the tendons and the surroundings of muscle fibers that we all see in our patients, sometimes with the skin as well. But much of the effect that we see in collagen 6, a little less in collagen 12, is actually on the muscle that is supported by the collagen 6. So the problem is in the support of muscle. Much of what we see clinically is that the muscle isn't happy with this abnormal matrix around it. Why that is important is because when we think about treating this, we have to treat the cell type in muscle that actually makes the extracellular matrix. And that, for the most part, is not the muscle cell. It is the extracellular matrix cells that are surrounding muscle, and they're called muscle fibroblasts or fibroblastic adipogenic progenitors. It's a different cell type that people in gene therapy haven't really thought about yet, but we have to think about it here. Sometimes the reserve cells of muscle, the satellite cells, also need collagen 6 to work properly, so that's a sub-problem that we may have to address. So one problem is the genetic mechanism. Second problem is the cell to target to really correct the disease if we think genetically. Keep that in mind. And the third problem, of course, is when do we treat, how we treat, what are we going to see if we treat, what can we expect when we treat, and that will be leaving up for the discussions that are to come. So my lab at the NIH is really trying to solve that problem of the dominant mutation, in particular how you interfere with the dominant toxic copy to kind of take it out of the equation, to eliminate it so that the normal copy can do a normal job. And that's done with several techniques that we can talk about as we go forward. There's one common variant in collagen 6, which is a pseudoexon mutation. I don't know whether anyone in the room has that. And that requires a separate approach called exon skipping, and we can talk about that as well. So that the main focus in my laboratory, including how to target these cells that make the collagen 6 as the main problems. So with that, I'm going to hand it to our first panelist, Russ Butterfield, to introduce who he is and what he does.
All right, I must have sat in the wrong chair to be first. But I'm Russ Butterfield. I'm a pediatric neurologist at the University of Utah, where I've been for the last 20 years or so, and been working in collagen 6 for about 10 years. And that we have a couple different interests in our laboratory in understanding genetics, first of all, and then understanding therapeutics, like how would we treat this disease, and some of the ways Dr. Bonnemann talked about. So first, on the genetics, one area we're really interested in is it's known that some patients with collagen 6 related disorders, and Carsten didn't talk a lot about the different Bethlem myopathy or congenital muscular dystrophy intermediate phenotypes that are more or less severe, but even with the exact same mutation, you can have any of those phenotypes. You can have a severe disease with a particular mutation or a milder disease with a particular mutation. And we're really interested in what are the genetic components of that variability. So what are the genes that might cause a person to have a milder disease, because that gives us some information about where we could have a target for therapies, right? That would be the idea behind that. To do studies like that, you need to collect large numbers of patients because those effects collectively are small. So we've been working with a group in Belgium to gather as many patients as we can find and get DNA samples and sort of a clinical thumbnail that would help define a person's progression over time and onset that would allow us to identify those genes. And that is a complicated process where we look at across all of the different genes, right, for an effect. So that's one area of our work. And the other areas in therapeutics, we're interested in how do we turn off that dominant negative gene. There's a lot of different mechanisms that one could use to do that. The one that we're working on in our lab uses a tool called ADAR. And if you've heard of CRISPR, CRISPR is a way to edit genes in the DNA. ADAR is a way to edit genes in the RNA. And so we take the transcript that comes off of that gene, and there's a way to edit that using a molecule called ADAR. We're still very early in those studies, just trying to get a sense for in a culture dish, can we make it work? So that's where we're at with that.
Thank you, Russ. So two important concepts. One is the concept of genetic modifiers. So when the same mutation that we know plays out differently in different individuals, every carrier, every patient is their own complex organism. And so sometimes the same variant in the collagen 6 gene will be either more severe or milder. And it will be very important to understand why that is, in particular if you have a person who has a mutation that should be severe, but is quite mild. You want to see what is the own body doing to rescue that mutation, because that could be a therapeutic target. And the other concept, of course, is that of gene editing. So you remember the flow of genetic information from the DNA, the chromosomes, to the RNA, and then making the protein. If you edit the RNA, that's read off the chromosome, it's not permanent, so you have to redo it. Whatever you do to the RNA, it's not permanent. If you go to the genes itself, the DNA, it's permanent, which sounds great, but is also doesn't leave room for error. So if you do something wrong there, you can't correct it back. So we keep that in mind. So I hand it over to Virginia.
Hello, hi, my name is Virginia Arechavala-Gomeza. I work currently in Spain, although I did work for many years in the UK. I've been working in neuromuscular disorders for the last 20 years or so. And in the last 5, I came back to collagen 6, which I had touched briefly in the past, but not full on. I lead a group that tries to design those gene therapies or genetic therapies, oligos or RNA therapies for different neuromuscular disorders. And one of my obsessions is to try to quantify what we do before we get into any kind of clinical trials. I've done it in the past for other proteins that have any different neuromuscular disorders. And while we are, again, developing different therapies for mutations, we're also using CRISPR-Cas or testing different routes to modify collagen. What we've been focusing the last 1.5 or 2 years is to quantify collagen itself in tissue culture. So you know that in general, when we are developing a drug in the lab, we tend to test everything first in cells from the patients to see if these things do give you a response. And we didn't have any way to quantify collagen from those patients. So this is what we've been doing. We've been trying to develop a method in which we can quantify not only the amount of collagen that you see in cells from patients, but also how disorganized it is. This is something that perhaps we could use it later on to stratify patients. We don't know yet because we've only done it in a small number of patients, but we know that we can see patients that have very disorganized protein, and then we can give it a number. And then we know that we've been able to also evaluate the effect of some of those drugs, again, in a petri dish, in the cells from those patients. So we have cultures from some colleagues that have been doing CRISPR-Cas on patients, and we have the cells before and after, and we can quantify that with a method. We've done this so then anyone can do it. So we're happy to share. Actually, the protocol has been published. We're happy to share it with anyone who's interested in doing it. Right now, we want to just keep on working on seeing if with this method we can perhaps check how the patients are, but most importantly, to be able to evaluate different therapies in a quicker way. As Carsten said, one of the problems is to target those particular cells that are the ones that are going to be producing collagen. So the other thing that we are very interested in is the delivery methods that we can use to get to those particular cells. We're not the only ones in this, and actually we're collaborating with a lot of people, but I think this is probably the biggest hurdle that we have right now, because we can cure cells in the lab, but we still need whatever is coming next to put those therapies into the right cells. So we're busy working with those cargos, so then whenever those delivery bands are ready, we are ready to go. But that's the other thing that we are collaborating with.
Thank you, Virginia. Again, a few very important concepts. One concept that is emerging here is that concept of genetic tools. You heard about ASO, you heard about CRISPR, you heard about editing. And the important and actually good concept here is that the tools that are developed can be applied across a whole bunch of different genetic disorders. That means that in the collagen 6 field, as small as we are, we don't have to reinvent every tool. We can actually apply tools that are being developed out there by many big groups and important indications that may rise higher to the intention of commercial interest and so on. But we can take advantage of that by using tools applied to our rare diseases as long as we understand the genetic mechanism that we want to fix. And so that tool, that platform concept is a very positive concept because it really accelerates research. And the second important point here is that we want to have things before the clinic, things before patients are exposed to any experimental therapies that tell us whether our therapy actually has a chance of working. And therefore the quantification of what you do to kind of compare head to head which one is actually better in the lab before you expose patients is absolutely essential, and you hear more of that as we go forward. And then you heard about the delivery problem again. That will come up many other times. That's a real bottleneck that we need to solve. And the last concept, I think that's, I would kind of say for everyone in the room here is that this is a very collaborative field. So collagen 6 researchers are talking all the time amongst each other and exchange methods, ideas, and reagents. So that's also good to accelerate things. Payam?
Hi, I'm Payam Mohassel. I'm a adult neurologist by training. I'm a physician scientist, and in my laboratory, I study a mouse model of collagen 6. I started that work about 11 years ago in Carsten's group, and I was at the NIH, and then I have taken that with me to Baltimore Johns Hopkins, where I am now. Our focus in collagen 6 research is a little bit more on the basic side, so this mouse model doesn't make any collagen 6 at all, and we're interested to understand what happens to muscle when there's no collagen 6 in the matrix. Why do the muscle fibers get weak? At the molecular level, what's happening, what pathways are involved. And so that's one aspect. We've identified abnormalities in certain signaling pathways, and we think that collagen 6 in the extracellular matrix is involved in regulating how those pathways are activated and deactivated. And when collagen 6 is not working right or is not there, those pathways kind of are stuck, and they can't really adjust themselves to the needs of the muscle tissues. So one of those pathways is the TGF-beta pathway, which our data has pointed us to. We found a similar thing in patient muscle biopsies when we studied that in Carsten's group, and also in the mouse model. So we're taking that forward and are testing that mouse with genetic crosses, or so crossing that mouse with mouse models that say have a deactivated TGF-beta in their muscles to see whether that improves the mouse weakness, or treating the mice with drugs that act on that pathway to see whether we can improve. So kind of identify pathways that are involved and then targeting them in a sort of a mutation-independent way. So it doesn't matter what the mutation is, what the cause is, can we downstream from that help the muscle tissue not be as damaged or recover better?
Yeah, thank you, Payam. So again, a few important concepts here. One is, again, the importance of models of the disease. Now, the mouse that Payam was referring to is a recessive model. It makes no collagen 6, but it is not a match to the human disease, actually. So the mouse is actually not that disturbed by not having collagen 6. You can measure that the mouse is weak and everything, but it's no match to a patient with loss of collagen 6. And still, it's a very important model, because models are only as good as the question you ask of them. You don't want, and you can't expect that a preclinical model is replicating everything in the human disease. That's never possible, with very few exceptions. But if you ask the right questions of a model, then you can get a lot of knowledge out of it. And so here, Payam is asking, what actually happens if you don't have collagen 6? What are the abnormalities that happen in muscle that I can possibly address with therapeutics? And so that is the so-called signaling. The matrix, the extracellular matrix around, regulates whatever gets to the muscle surface, including molecules that talk to the muscle, the signaling molecules. And what Payam found is that there is a dysregulation of that you could possibly address with medications that may be developed for something completely different, but that addresses the same signaling molecules. So that's repurposing of medications that are already out there that can be repurposed perhaps for the purpose of this problem. So that's the importance of understanding not only the genetics but also the mechanisms, what happens after the genetic mutation. And when you think forward into therapeutics, this is going to be a problem that you will not be able to address with just one therapeutic alone. I think you want to have multiple levels of attack from clinical care to gene editing and everything in between. And that's why these mechanisms are so important. Franziska.
Yeah, hi, my name is Franziska Haarich. I'm a postdoc at the University Hospital Schleswig-Holstein in Lübeck, northern Germany.
Where they have excellent marzipan.
That's true.
Yes.
Yeah, we're famous for that. Yeah, I started in 2019 with my PhD under the supervision of Janet Erdmann, who was affected by collagen 6 as well on her own, and she started doing research on that some years before that, and I started with that in my PhD, and it was really special project in that way, and the aim of my project, and I'm still working on that as a postdoc, is to find something as a therapy approach that does not just target that one mutation that somebody has, but multiple. And I'm working on the dominant form, and I'm looking for other target sites on that gene so that I can, with that, regulate this gene down without affecting the wild type or the healthy allele, so that can still work and work properly, but the mutated one is not interfering. And I want to do that, so targeting another site so that I can, it doesn't matter what kind of mutation it actually is, it just has to be a dominant mutation, it has to be on the same gene. So it, for example, collagen 6A1 or A2 or 3, and then I could target them all with the same stuff. That's the overall idea, and as everybody said, the delivery is difficult, especially in the human being, but it can also be difficult in cells already. So there are quite some bottlenecks, and I had to fight for some years to get it into the cells and to get this idea working, and now it's working. Not that efficient as I want, but there are always things that you can do to be more efficient at the end.
Big shout out to Janet Erdmann, who we tragically lost. And she was just the most amazing individual. She was affected by collagen 6. She was a really top-notch cardiogeneticist, so she publishes in Nature and all of these, and figured out her own disease mutation in her own genetic research and found that she has collagen 6 and redirected her research to collagen 6. A most amazing individual and a huge loss to our field. Second concept is what Franziska mentioned with another use of CRISPR. You heard about CRISPR as a gene scissors, but CRISPR can do so much more. If you think about CRISPR, at the basis of it is a search mechanism, like in your word processor, when you have a novel and you're looking for one particular word or spot in that model, you can do, Apple find or whatever, you can do control find, type in that, and the word processor will jump right to that point. That's what CRISPR does as well with the guide. You can direct it to any spot in the genome with high precision. And what you deliver is, again, the toolbox that you can do. You can either cut, you can edit, or you can regulate, as Franziska is trying to do. So she kind of gets, with the CRISPR machinery, right in front of the mutated chain and turns it down without cutting the DNA. So that is really making really good use of genetic tools where you can get to a point where you want to, and then you can regulate the gene down so that it's shut off without cutting in the DNA. So if that works in the end, that'll be really cool. Vittoria.
Good morning, my name is Vittoria Cenni. I come from Bologna, Italy, and I study, I've been working on muscular dystrophies for 20 years. And in this period, in the last 6 years, I began to study collagen 6 pathologies. In particular, I studied tendons, which are the organs that connect muscle to bones and allows the movement of bones following the contraction of muscles. Tendons in collagen 6 patients are impaired, and however, if in a first period they thought that these impairments were due fundamentally to the reduction of the contraction of the muscle, the nearby muscles, now we know that tendons suffer from primary defects. In fact, in my lab, we study tendon cells, and we have discovered that tendon suffers from alteration to the response of mechanical strain, to alteration of the response to TGF-beta, and also to the impaired polarity induced by migration. So in my lab, we study the pathogenetic defects of tendon cells that can help us to find novel targets to discover therapeutic targets and to improve their response to these therapeutics.
Thank you. That is another very, very important concept in our disease group, which is that this is not a pure muscle disease, it's also an extracellular matrix disease, a connective tissue disease. And so it is a disease that affects muscle as a function of the abnormal extracellular matrix, but the other cells that really make a lot of collagen 6 and are needed are the tendon cells, so the tendon that attach muscle to bone. And without a tendon, you wouldn't be able to move because the muscle needs to transmit force onto the bone to move. Now, the classic diseases of the tendons and the connective tissues are Ehlers-Danlos syndrome. And the classic diseases of muscle are muscular dystrophies. And so what we have here in this room is a hybrid of the two. So we have a muscle disease with Ehlers-Danlos features because the tendons are involved as well and an important part of the disease. And also sometimes, like in collagen 12, you have more tendon pathology, but then a secondary muscle pathology as well. So it's kind of muscular dystrophy with EDS or EDS with myopathy. That's kind of this spectrum of disorders that we have to address at the same time. So from looking at your family members that this is playing out in front of you with both the hyperlaxity and the bendiness of the joints, but also these contractures, the shortening of the tendons that can be really causing their own problems with mobility and disease progression. And if we ignore them, we ignore half of the disease. So thank you, Vittoria, for doing this. Jeff.
Thanks, Carsten. So my name's Jeff Molkentin. I'm a scientist at Cincinnati Children's. I've been there 30 years running my own lab. I've been on the SABs of a number of different companies, including companies that are going after Duchenne by CRISPR. And I'm still working with one of the companies that got bought by a bigger company. And their goal is to use CRISPR where possible for Duchenne. And there you only have to target the fiber, the muscle fiber. Unfortunately, we all probably need to target the fibroblast. So my lab ends up being an expert in fibroblasts. We've been working on it for 25, 30 years, and how it regulates and controls the extracellular matrix in the basement membrane. So given our 20 plus years of work in Duchenne, my understanding of kind of the entire biotech kind of world around muscular dystrophy and the extracellular matrix and the fibroblast, a knowledgeable family came to me whose husband is a biologist but also is a venture capitalist, and he works on setting up biotech companies. And he basically, in a way, hired my lab to try and figure out a way to either redesign, reconfigure, or fix the collagen 6A2 gene in fibroblasts. His son is suffering, he's homozygous recessive, the same mutation. And so my lab's big. We have a lot of different tools. We use viruses, we do gene therapy, we study the basic biology behind TGF-beta signaling, fibroblasts, And so we went all in, and we are trying everything we can think of. Two things that we can count, at least from what we've done recently, we've used from Scholar Rock their clinical TGF-beta inhibitory monoclonal. There's a mouse version of that. We've used that and treated our collagen 6 deficient mice, the same ones that you guys had, the 6A2 nulls, and it didn't help at all. So TGF-beta is overactive, there's no doubt about it, we see it. Those mice suffer, and in the process, TGF-beta becomes activated as the muscle atrophies. That also impacts the tendons. If we block that, it doesn't do anything. The muscle is still bad. Another really bad feature is the neuromuscular junction. That's a specialization within the ECM that the motor neuron touches. Those end plates are defective in most of the Ehrlich and Bethlem myopathies. We've gone in and tried to re-engineer those as well and make them bigger. There's a way you can do this with a protein called DOX7. When we enhance the neuromuscular junction in our collagen 6 null mice, it makes them worse, which is really hard to believe, but it's probably not telling us that it's the defect there. It's the neuromuscular junction is responding to the extreme atrophy that's occurring in the disease. And it's secondary to that. If we beef up the neuromuscular junction, it actually makes the mouse worse. So we're going at this systematically. We're taking each thing, we're taking it off, and we're trying to find a way to really re-engineer what collagen 6 does. So what we've done more recently in the last year is we used AI to predict everything collagen 6 interacts with in the basement membrane and ECM. It's an incredibly simple protein in a way because it has one major domain. It's a von Willebrand factor domain, a VWF. It's a repetitive mesh. It's almost as if you threw a fishing net out into the basement membrane. And it's the same consistency throughout. And it binds to a bunch of different things. And the fibroblast is what makes that. So if you're going to genetically address this, you've got to get to the fibroblast. What we're going to try and do is use the myofiber to address what that fishing net's supposed to do. It's supposed to hold everything together. It's supposed to make sure the porosity of that ECM is correct. We're re-engineering that because we can get to the myofiber, and we can do gene therapy to the myofiber, and we want to just rebuild what collagen 6 normally does through that VWF domain from the level of the myofiber. And that's what my lab has really put a ton of energy into more recently, and I think we're close on a few things that might really potentially do this. It'll also work with the dominant negative, which probably is part of the same loss of interaction. I think you've noted that in the very beginning when you found those mutations. It's kind of the same idea. So my lab is all in. We're trying to think of everything we can do to go after this disease. We're still trying to find a way to do gene therapy with various AAV constructs for the fibroblast. That's really, really hard. I'm not sure that that's going to be the virus to ever do this. I think we need a different mechanism. I don't think AAV is it, but we're working on that as well. We're working on the loss of tension. Is it just that? We're working on probably everything we can think of because we want to stop this disease. It's horrible. I mean, watching this family and their son, who's now 3.5, slowly kind of wither away. It's heartbreaking. And we're smart enough now as a community. We've done enough genetics. We've done enough gene therapy. We've done this stuff. We should be able to figure this out. And watching the mistakes that's happened in the Duchenne field, we can do better. And I think we just need the resources and time, and we can do this.
Yeah, thanks, Jeff. So it's the concept here is really that we are extremely lucky to have a laboratory like Jeff's laboratory with all the experience and resources involved in our field. And you can see how creative thinking around the problems to really kind of address it from unusual angles and really take experiences in other diseases and other problems and apply them to this disease is really a very creative way forward. And you never know where the breakthrough will happen, and so all of that has to be supported. And the other thing, of course, is to pull in support from wherever we can get it, and these are, of course, difficult times in terms of research support, and everything that we can do to support the idea that research is important to address these disorders will be helpful. Daniel.
Hi everyone. So my name is Dan Moore. I'm a postdoctoral fellow in the lab of Severio Tedesco in UCL and the Francis Crick Institute based in London. And so the sort of broad focus of our lab is to build human with human cells, 3D muscle models that we can use in the dish to assess different phenotypes from different muscular dystrophies. So I have to say that I actually don't personally work on collagen 6. I'm substituting in for a colleague today who's done a lot of work on this. But what we're hoping is that broadly by building these models, we can start to assess functional readouts before we go into a mouse model or any other type of preclinical model. And so hopefully that will give us sort of a more accurate assessment of the effectiveness of any potential treatments. And we also hope to be able to use these models to begin to look at more basic disease mechanisms in different contexts. So that's kind of broadly what we do. And any questions on that, I'm happy to answer.
Again, the concept of having really good preclinical models that are allow for relatively rapid testing of different approaches, head-to-head comparison of approaches. And these models are, of course, human-derived models. So they're actually human cells that may be derived from patients, and they're made into, via this IPS process, they're made into stem cell-like things, and then they're driven to differentiate into the cell type that you want, all from the same parental cell line from the patient. So you can actually rebuild a little organ in the dish, a little kind of micro muscle in the dish, and you can see how it physically acts, how it forms a matrix, how it transmits force, and how it can be corrected or not with the therapies that we use. So all of these tools are really useful. Well, thank you all. We have lots of discussion prompts here at the slides, but we're not going to necessarily adhere to them, at least unless you want to. And now we're going to open it up for questions. You can ask general questions or specific questions to any of the researchers here in the room. And is someone with a microphone out there? Oh, yes, perfect. Can you use a microphone, you think?
Okay. Does anybody else have one? We'll let Keith think about that for a second. Open question? Absolutely.
Our daughter specifically has the intron 11 for Cal6A1. So I was just wondering, maybe you already touched on it with just the basic outline, but just wondering if there's anything specific related to that research that you could touch base on for us.
Yeah, so that, to bring everyone up to speed on that one, that was actually discovered in patients some years ago, where we as clinicians said, well, this patient has Ullrich congenital muscular dystrophy, full stop. If this is not true, I'm going to retire and open a coffee shop or something else. And so we had several of these patients in London at UCL with Franziska Haarich in our group, but we couldn't find with whole exome sequencing, we couldn't find what was going on. There was no diagnosis made genetically. And then that was the beginning of a next phase of next generation sequencing called RNA sequencing, where you take the transcript, the RNA from muscle, and sequence that and see maybe something is happening that's hidden from our regular genetic testing. And so it was. There was a variant, a genetic change deep in between the exons that we look for mutations in this intron material in between that typically spliced out, kind of got rid of when we do the transcript that forms the protein. But what it did, it activated part of this quote-unquote junk DNA to be now part of the protein. We'll call that a pseudoexon or poison exon that is now spliced into the protein and causes this toxic dominant negative effect just by the virtue of being there, being included there. And what was really quite surprising is after this initial 2 patients were discovered with this method, it turns out this is one of the most common single mutations in collagen 6 that went completely under the radar, not discovered because it was deep intronic. And we now have over 50 patients that I know of with this, this variant. And Reghan Foley, who comes, I think, later, just put out a beautiful paper on the natural history of this variant. Why is this important? So sometimes when you find something with this precision diagnostic, it actually comes with a therapeutic idea delivered to you on the platter, which is exon skipping. And you may have heard of exon skipping in Duchenne muscular dystrophy, where you do exon skipping to That gets a little technical, but when you skip something in Duchenne muscular dystrophy, you can actually force the transcript, the messenger RNA, to be back in frame by taking away more than what's missing in the patient. So the patient has a loss of genetic material but makes no protein. If you skip another exon, take a little bit more, you can actually reframe it, make protein again. An abnormal protein, but yet a protein. If you have a mutation that is a pseudoexon, poisonous mutation, if you skip that, you generate a normal transcript because you just take that out and it becomes no longer interfering with the transcript. So it's something that is extremely enticing as a therapeutic approach. Where do we stand? So we and others have developed ASOs, so small kind of molecular probes that do this exon skipping, and that works very beautifully in the dish. So when you do this in cells in the dish, you can really absolutely correct it. But here we're now facing the delivery problem. We have to get these ASO effectively to these fibroblasts in muscle, and we have generated a mouse with this mutation that has the human context there, and so we can use that. And we're kind of slowly inching towards addressing that by targeting the fibroblasts. I think we'll get there. Not quite there yet, but so the therapeutic proof of principle is ironclad, it really works. But to get it into a living organism, into the muscle and all the fibroblasts, that is still the bottleneck that we're all trying to address.
Thanks for being patient as I waddled around.
You made it. Hi, I'm Inbar Sharon. I have 3.5-year-old twins with a dominant negative mutation in COL6A1. Sorry, I apologize if this meanders a little bit, but one of the hardest things, my twins were diagnosed last year, one of the hardest things was, as a parent, is tempering expectations around developments in research and therapies. So I'm just curious from all of you, kind of in your work, where do you think we'll be in 1 year versus 5 years? Like what are the timelines that we're looking at? And especially with the acceleration of AI and additional tools that can be integrated that might, speed things up, hopefully. What does that look like?
Who wants to take this?
I'll give it a start by telling a different story. So when I started in this game, which is a little over a decade ago, we had no therapies for any of the disorders we treated. And I see patients with all types of neuromuscular disease. And the only prescription I ever wrote was for prednisone for muscular Duchenne. Now, fast forward a decade, we have multiple types of exon skipping therapies, which are a little genetic trick, or just gene replacement therapies. We have made a lot of progress in Duchenne and SMA and other spaces. And if you were to have come to this meeting 10 years ago, we would be talking a lot about the mechanism, like how does collagen 6 work? And we wouldn't be talking very much at all about therapies. But what I think you see in this group is that about half of us talked about therapies. So I think that's really hopeful, but I don't know in 1 year or 5 years, but I think that transition happens already and it's accelerating. So I don't know to give it a timeline, maybe Carsten or others can do that, but it's moving quickly as science goes.
I am a researcher that I've made my career of doing genetic therapies, but I see now, like in things like Duchenne muscular dystrophy, there are like many different options for patients, but also none of them is completely curative yet, but many of the ones that are coming also are those that are not gene therapies, not RNA therapies, that they are maintaining the muscle better, that they are keeping the patients healthy for longer. So while we are doing this research, we're learning in many different aspects of the disease, even when we are focusing on a particular way of treating the patient. So what I want to say is that you shouldn't really put your hope in a particular kind of therapy, because there are lots of different avenues that we are testing. And sometimes things such as physiotherapy and keeping your kid healthy up till the moment when that better therapy is arriving, it will probably do more for them than any of the things that we may be working on now. Many of the things that we're doing now will not work, and that's the fact. But I think knowing when something doesn't work lets you learn from that experience and also apply that to the new things that they are doing. And I think it's true that 15 years ago there was nothing, and now there are many different things that people can do. So I don't want to give false hope, but it's true that there are things coming, and not always from where you were expecting to see them.
Anyone else? I mean, it really is. So I think one of the hopeful things is that we are not alone with the collagen 6 disorder, not even with muscle disease. So whenever there's a breakthrough somewhere else, we can learn from that breakthrough and see how we can apply it. And once we do the first, even if it's a single patient therapy in a collagen 6 patient that is perhaps changing the disease, that opens the entire field for the next one and the next mutation and the next approach. So it really has an accelerating effect. Once we get there, it kind of can open an entire new era of therapeutics. And it may start, as you mentioned, somewhere else, and then but immediately be applicable to what we do and kind of accelerate that. So I think it's very encouraging that many of the approaches that are now being pursued in genetic medicine are not disease-focused alone. They're actually applicable to many, many other conditions. And as long as we take advantage of that and do our own research and bring it together, that could make a, I don't want to say quantum leap, but can kind of all of a sudden from kind of not moving, get the field going. We always want to do that in a way that is safe and responsible so we want to kind of put only our preclinical models at risk, but never the patients. Other questions?
Hi. So I hear a lot about the difficulty of delivering the genetic therapy. So like if there was a cure, I understand the concept, but can you help me like more visualize what that really means? Does that mean you like if you deliver it in someone's arm, it only goes like within 2 inches?
Yeah.
Is that like it's going just to your legs, or
no? That's
you know
a really
what I mean?
good question. So let me start on this and then have others also. So one of the challenges, if you will, of dealing with muscle disease is that muscle is one of our largest organs. So depending on how fit you are, part of your 50 or 60% of your body weight is that one organ, muscle. It's only one muscle, but it's 650 individual muscles that all play roles in movement and expression in survival. And you want, with the therapy, you want to get to all of them. So just injecting it in one muscle is maybe not the greatest idea if you really want to help the patient. I mean, sometimes used for proof of concept, can you do something there? But you're right, I mean, just kind of putting it in the arm, in the muscle there, that will not be what we want. And so for systemic delivery, to get to all muscles, our best bet is to take advantage of the bloodstream, to inject it into the vein, and the vein goes back to the heart, and the heart pumps it through all the muscles in the body. So that comes with 2 caveats. One is when you put something into the vein, it first goes into the liver. And the liver is really good at taking out things out of your bloodstream. And so you want to get past the liver into the organ that you want to address. And then within the organ that you want to address, you need to get out of the vein, into the muscle, into the right cell types and that you want to actually address. And so in our case, it's the fibroblasts, maybe also the stem cells of muscle that you want to get to. So you need to develop a tool that is capable, venous injection, to get through the liver, not being taken out, don't damage the liver, and get to the muscle, out of the blood vessels, into the right cell type. And you can do that with a number of different tricks. And I ask our research, what are some of the tricks you can do there?
well, the delivery is one of the things that I've been more interested in, and it's not just myself. I've been leading a network of researchers that that are looking for delivery options for different therapies, such as those. And the good news is that that's a common problem. That's not something that is only collagen 6 disorders or muscular disorders. It's a problem for many of us. I know that may sound counterintuitive, but actually having the same problem that people perhaps having cancer or other disorders is good for us, because then there are many more resources working on that. That depends a lot, as we've mentioned before, there are lots of different possible ways of treating this disease, and then they could be divided roughly into things. Like if you want to give large things, sometimes you need to use a large vessel to take that into whatever you're going to take it. And we may be thinking something like gene therapy or so on, which uses viruses. If you want to go for smaller things, sometimes you may go for something like nanoparticles or lipid nanoparticles. You know, if you think of like the COVID vaccines, quite a lot of the success of that vaccine that was based on RNA, it was not just on the way the RNA was designed, but also on all the formulation that allow it not to get to the place where it had to act. So I think there are lots of things that are being studied, and until now we have to get ready those cargoes. So then whenever that new molecule is ready, we are ready to go. Right now, if you're thinking in this kind of small molecules for rare diseases, that ones that work better are the ones for muscle, for tissues that are accessible, such as the eye or the CNS, if you go straight into it. So, and the liver, because as Carsten said, everything goes to the liver. So most of those drugs are being designed for this kind of research. But even though they're not for us, they are learning so much by developing them that it's something that we can also take advantage of, because they are developing the techniques, they're developing the manufacturing system. So I think by the time we can overcome some of the hurdles we have, we will have all those other disorders that would have been advanced in the field. It's not, we don't work in separate worlds from everybody else. So the field is advancing, and once we have it ready, we will be ready to take advantage of it.
So for this disorder, we're really stuck with how are we going to genetically modify the fibroblast? And there are a lot of other diseases of the fibroblast, like Ehlers-Danlos syndrome, where we've known the mutations and we've known the genes for years. But as a field, we haven't had an effective delivery method in vivo to get genetic material in, or even antisense oligonucleotides into fibroblasts in vivo. It's a really hard call. People are working on that. We're going to solve it. This is a more exciting time in science than I've ever seen in my life. Things are moving so rapidly now. You know, just in the last several years, we've discovered whole new ways of modifying genetic material with CRISPR, and things then related to that. So the field is moving incredibly fast. We need to find how to target that cell type. Now, for this disease, there may be a surrogate. If we could target the muscle itself, the myofiber, that's a cell type. We already know a couple vectors that'll do that and do it quite well. They use it in Duchenne. So the AAV vector, there are companies built around optimizing that vector so that it goes in the muscle even better. What's currently on the market isn't the best. And you guys have probably heard with Sarepta, the problem they had with microdystrophin, getting that into the Duchenne kids. And then the way in which they do this is right at the limit of toxicity. They are at such a high titer of this AAV that it basically almost, makes these kids extremely sick or even kills some. That's the level of virus they need. Now, what's happening now is the technology from 5 years ago. That vector, that AAV, is getting much better. The companies that are now next ready to go with this, I think are going to have a much easier time. So then for this disease, can we use the myofiber to fix what the fibroblast didn't do right? And I think there's a way to do that as well. So we have two ways. We have to get into the fibroblast somehow, or we have to find a surrogate, and the best one in my mind is the myofiber, because it's what touches that surface. And it certainly can make collagen-6 developmentally. Most people don't realize this, but like in the very immature state, the satellite cell, the stem cell, turns into a myoblast. That then turns into the myofiber. In the very beginning, that cell makes collagen-6. It has to, because it's right there next to it. But it shuts off and it never comes back. So you can't use it to make collagen-6. So, but you could use it to make something else. So we have two ways to get in, and I think now is the time. Things are going so quickly. I feel really excited that in 5 years, we can crack this one.
Yeah, and to give you an example how this is being done, Jeff mentioned in the muscle AAV field, we now have AAVs, which are viral capsids, where we put the therapeutic gene into. They're basically a virus envelope, which is protein, that packages the genetic tool, which is either a gene replacement or a CRISPR tool, but it's packaged into a viral capsid that looks like a little virus to the body. And what the muscle field and also some of the central nervous system field has done is to change the envelope, the surface of the virus, in a way that it now homes into cells and recognizes them specifically. Every cell has things on their surface that are specific to the cell. And if you give the virus or the ASO a homing device to recognize that, dock to it and go into it, then you have a way into it. And that's been successful in the muscle field with these new AAVs that are homing to muscle much more effectively, and you can have lower doses and better effect. And the same arguably could also be done for the fibroblast by just looking for things that recognize docking stations on the fibroblast and homing devices on the fibroblast to get there more effectively. And that's done now with really very modern tool sets where you can screen huge libraries of potential variations of these viruses and see which ones come out as the winner there. That is something that wasn't possible 5-10 years ago, but now is, and I think that's one of the ways forward. Yes, here. The microphone's coming.
Hello, Jason Johnson. My daughter Emmeline she goes to Cincy Children's, but she's been to see Dr. Bonnemann at the NIH a couple times as well. And I know when we've gone there, we've always left with the real sense of pride that this country prioritized research and medicine to solve problems for the greater good. And it's these last 2 bullet points I'd like to hear about from this community, because I'm hearing a lot of optimism from you guys, but it's hard when you read the headlines about all the funding cuts and the sort of attack on research, which seems to make no sense. I'm just curious what constraints that's causing for you guys and what we as a community can do to help support you.
Jeff, you're up.
So I'm also at Cincinnati Children's. We have an incredible research foundation. It ends up being probably one of the largest pediatric research institutions in the world, arguably with Boston Children's. And this thing that's happened in the last 6 months with the assault on research on almost every level, visas for our workers, it's fundamental education changes, that science is bad, that somehow it's an evil thing, and then just directly cutting and pulling funding from NIH sources to cripple universities, not to hurt the research, but to hurt the universities. This is a fundamental assault on what we're trying to do. We're trying to help mankind. I feel really strongly about this because I feel like this is the last thing we do best in America. We are the best biomedical researchers, the most ingenious. We're by far one of the most innovative countries in the world. I mean, look at everything else we've done. Why would you cut off from this boilerplate of innovation the very source of it, which is your academic centers? Why pull funding? Why take the NIH as the method to hurt the universities? Why take away what we do best, what we lead the world in? People come here from all over the world to do research, postdocs, graduate students, because we're the best. We still lead the world. We cannot, as citizens, allow our politicians to disregard medical research. This is too important. It's such an exciting time. We're doing more now than we've ever done before. We are on the precipice of changing so many diseases across the board. If they cut us off now, you guys, it is going to be a disaster because you can't get this stuff back. You can't just say, okay, well, in 4 years we can reinvigorate this. We're going to lose more than a decade if we try to get this back in 4 years. We need to keep this going.
Amen.
Let me just add by the way, I'm Rob Sunrus. I'm president of the board for CMD. So thank you all for coming. And, you know, we work a lot with a bunch of different organizations funding both overseas and here in the States, right? So I'd have like two main kind of points to that. One is advocacy is really important. I think we have a couple sessions maybe on talking about advocacy, but it's getting that message to, you know, your congressmen, your senators, et cetera, and really understanding. Rare diseases used to be bipartisan, and for some reason we're off kilter right now. It used to be something everybody supported. So advocacy is really big. And then the second thing is funding. Obviously, you know, everybody gets funding from different sources, and, you know, some of those come from private equity, some of them come from private grants, some of them come from grants from nonprofits like CMD and the other organizations that are here. So that doesn't necessarily impact, you know, the things going on at the federal government level, but from a continuing to help push that research better, that's where the funding really comes in. I know we've heard stories of Duchenne and SMA, and I think they've had we at CMD are proud we've had about $4 million in grants that we've been able to support over the years, but that pales in comparison to what the Duchenne and SMA community can do. So, and it's just a numbers game. So, and, you know, as you see us doing fundraisers and stuff, it all kind of goes back around to help some of this stuff. Russ?
And I'll just add that your voice is really important in communication in the political sphere and in funding. There's a real limitation to some things that we can say. Yep. You know, I can't be, and it's really sad to say, I can't be as strong an advocate as I would like to be because maybe my university will lose all funding.
Yep.
And I know Dr. Bonnemann's directly there at NIH and probably can't say the things he would like to say. Yeah, right. So, and I'm probably more insulated from that at the University of Utah, but we still have these political spheres that we have to navigate, and your voice becomes way more important when our voice gets muffled like that. Yep. So we need you to speak.
What I will say, last week the Senate Appropriations Committee was looking at the budget, and there was actually strong bipartisan support to not cut the NIH budget and increase it a little bit, meaning there is still a consensus there, but there is kind of less eagerness to be vocal about it. If you encourage everyone that you know to be vocal about it, then there is no longer the fear of speaking about it. I think the basic intent is there and the bipartisan support is there, but you want to kind of overcome that climate where you feel you can't say certain things anymore by encouraging your representatives to be vocal about it. I think that would be, irrespective of party, I think that would be a huge step.
Agreed.
One other thing, maybe just to add on the advocacy, that right now at a federal level, there's a thing called the Action Plan Muscular Dystrophy Coordinating Committee at a federal level, combines efforts from FDA and NIH and the CDC, and they set the agenda, the research agenda for the next decade, and that process is now in public comment period. So if there's things you think are important, this is a good time to make your voice heard in these public comment spaces, and I can try to find a way to get you a link to be able to do that. But that's an important, just by a natural 10-year cycle, that's up for discussion right now.
Hi, physical therapist here. In light of hopefully forging more research forward, In trying to investigate and learn more about collagen 6 myopathies, initially we turned to the resources that we have, the literature available. Looking into etiologies, my brain sparked ideas about things that work on tendinopathies in terms of reorganization of the fibroblasts, things like vibration therapy, things like initially even eccentrics, although I've come to now learn a little bit more about not doing that. However, when we look to the research, most of the things that we see are not collagen 6 specific. They're painted across all myopathies and usually even just remnants from Duchenne's dystrophy, which isn't the same thing, obviously. So how can the people on the ground, like us, with the patients, when we have these ideas or we think, hey, I wonder if this could work, we're trying to get them to the finish line, where the finish line being hopefully something wonderful coming from your guys' laboratories and clinics. But when we're there every day with the patients and we're wondering, what are the actual things we can do with these people every week? Where do we go? How do we know what limitations are? How do we know it's worth trying or what's not worth trying?
Yeah, it's a really good point, and I take the first stop at it as a clinician. So, I mean, there is, you're right, much of what's known in many of the neuromuscular care situations comes from the most common neuromuscular diseases, meaning Duchenne and SMA, which has its good and problematic parts to it. It's good, that certain principles of neuromuscular care certainly apply across all neuromuscular conditions, but the drawback is, of course, to make everything one disease when it is not, and the disease-specific problems are not really proactively addressed enough. And that is really a pitch to us as clinicians to be more more clear about what the guidelines are that are specific to these conditions. Now, it's not hopeless because we have QCMD, where there is a website where you find care guidelines and disease-specific care guidelines. However, I'm the first to admit that some of them are really old. They're kind of 10 years old, but we're making progress. So we started just this year to update the care guidelines for marathon, Lama 2, with a meeting at ENMC. And it's a European Neuromuscular Center. It's kind of a small working group in Amsterdam, internationally sourced, where people sit in the room and try to come up with new guidelines and put them out there for everyone to access, including the caregivers, so that when you come to clinic and you say, well, I have this disease, I don't expect you to know it, but please go to the website and look at what other experts have recommended. It's important for that. And so QCMD will again play a pivotal role in distributing that across the world. And then it's really is making sure that the info gets out there, that these are available for free and for access, and for, of course, patients and families to be vocal and to say this is where it is, and for the care providers to not be miffed if the patient knows more about the disease than they do, which is sometimes the case, but be a partner in the care. Other?
I'll just maybe say, note your role as a physical therapist, but I think of the role of our therapists in clinic, who are very expert in neuromuscular disease, and they work on the clinical trials, and they do lots of different measures and have a different role. They're not the ones actually working with the patients every day. In Utah, my patients might travel 2 or 3 hours, so they have a therapist close by. And what I've seen amongst those therapists, that some really engage and some don't. So just even your interest and presence here helps to engage in the idea of like what works, what doesn't work. And we really need that engagement. I think a lot of therapists are trained in sort of an orthopedic model where you get your knee surgery and then you get better, and they struggle when we have a progressive disorder. It's like, what do we do? But I think the ones we've had the best success with are the ones just willing to engage and communicate with us and have a conversation.
Yeah, and I think the registries are key. The QCMD, CMDIR is key, and it is a patient-facing registry. And there's certain functionalities that can still be built into where you can kind of crowdsource outcome measures and crowdsource experiences with the disease and certain interventions. Not everything can be done in a controlled environment in the single center. Sometimes it's really the things that happen in the community, some observations that are important and shouldn't be missed. And registries are a place to put that also. I want to ask some tough questions here of my panel, and this is in point 3 is what are reasonable therapeutic expectations? And that's that's an elephant in the room. You know that in some disorders like spinal muscular atrophy, when you miss that window of opportunity right at the diagnosis before the disease even starts, that's when you have your impact with the therapies, and everything after that is really getting much worse. What can we expect in collagen 6 disorders, collagen 12 disorders? Do we have a narrow window of opportunity? Can we expect that things get better? How long is it going to take? What are the expectations? The hard questions here. Who wants to take them on? Including physicians in the audience. Payam, yes, I knew you would.
I can start. So, of course, it's a challenging question to answer definitively, but clearly, as diseases progress, things change in the muscle, for example. So if you look at a muscle of a patient who has collagen 6 in the first couple of years of life versus when they're 20 or 30, it's going to look very different. And we can see that just by doing, for example, MRIs or ultrasounds. And probably the treatments that are going to work in that 1-year-old or 2-year-old may not work in someone who is much older. And that's probably true for nerve diseases like SMA. There's clearly an age effect. And sometimes, as Carsten mentioned, if you are past that point, the treatments start to lose their efficacy. Now, muscle has one advantage that, say, the nervous system otherwise doesn't, and that's its capacity to regenerate normally. So if there are ways that we can harness that or reprogram that or reactivate that, that will probably allow us to improve things. Also, it is, there are groups that are working on cell-based therapies. This is a little bit, again, much farther from coming to the clinic. There's a lot of problems that need to be solved, but those actually don't depend on what disease is affecting the muscle. It could be a chronic inflammatory disease of the muscle, myositis. But if you can kind of put stem cells in there and rebuild muscle, you can recover the function potentially. So there's, as the disease changes over time, our treatment approaches may also change. And hopefully in the future, we will have, say, 10 different treatments, and we know 1 and 2 work early on, 3 and 4 work in the middle, 5 and 6 and work later in the disease, and you will be able to kind of have your options as you go through and weigh benefits and risks for each individual. Anyways, that's how I think about it, and I think that's another reason to pursue different modalities of therapy, because again, gene therapy and targeting the fibroblasts is really important if you want to cure the disease early on, but that may not help someone who's had the disease for 2 decades, 3 decades.
Yeah, that Jeff, muscle as a regenerative tissue. What's the biologist's view?
The skeletal muscle is a remarkable tissue. I mean, it's highly regenerative, and this disease, though, has very little regeneration naturally occurring. Duchenne muscular dystrophy, it's incredible. Almost 20% of the fibers at any one time in a young child with Duchenne is actively undergoing some level of regeneration. And in this disorder, it's very low, almost none. So but it doesn't mean you can't use a regenerative approach. It's just not part of the normal disease course. This is not a disease of ongoing necrosis. This is a loss of muscle force, it's a loss of muscle size, it's an atrophy. And so we could potentially still go after this with some kind of a rejuvenating cell source like muscle stem cells, or reawakening the ones that are already there with growth stimuli, I'm not sure that's been tried. I think it's worked a little bit in the
Right, yeah.
LAMA deficiencies.
Yeah, and we are now learning that, you mentioned it before, that the immature muscle, so the muscle stem cell niche is actually needs collagen 6 as well. That's the one part of muscle that does need it. And so that niche is probably affected by the disease as well and not as performative as it should be in regeneration, but that gives us a niche to address as well.
That's a great point. I mean, I don't think the field has addressed this yet. My lab is now actively working on, can we take the stem cells that are already there, and Carsten's right, that they do like to have collagen 6 around them. They sit in these little niches, and almost 5% of your total nuclei, if you're a child, in your fiber are these stem cells. So it's really quite remarkable how many you have. Can we reawaken them? Even though they maybe don't have the right surroundings, we should be able to, well, we're trying to do that. We think we could get them going again, help them build muscle, because first and foremost, your paper back in 2006, I think, you called this an atrophic myopathy. And that's fundamentally what it is.
Yeah, that was looking at the very, very early muscle biopsies where we didn't see the degeneration, regeneration, we just saw shrinking fibers, yeah.
Yeah, so I think we have to maybe think about that a little bit too.
Right, I just remember we had a paper on transcriptome analysis a couple of years ago that in that recessive form of disease, we didn't see signals tied to regeneration or tied to replication of DNA even specifically. So, and we've never followed that up, but that was one of the key signals we saw in those cells.
So the muscle acts immature. So it maintains more of a fetal-like program. So when you look at the transcriptomics, you guys have all published this too, it looks like it's never fully had a load. It's like if you go into outer space and you're an astronaut, right, your muscle shrinks because it's unloaded. This muscle in these kids, and adults, it looks like they're unloaded. Like
That's
they were in
exactly
outer
right.
space.
That's exactly right. Same for collagen 12, by the way.
Yep.
By the way, you already get a sense that we're kind of defining different targets that you can address, and I think the future of therapies is to not just kind of put all eggs in one basket, but really do multimodal therapies on different ways you can address this disease at the same time. Most cancer therapies, if you will, are usually not just one drug. They're kind of multimodal therapies. And of course, this is not cancer, but it's a complex disease that plays out in different ways in different cells that all could be addressed as part of the therapeutic concept. One thing that's also, I think, a somewhat hopeful observation totally from a different disease category is if you look at the field of fibrosis, which is the buildup of connective tissue in organs. There's lung fibrosis, liver fibrosis, kidney fibrosis that drives different diseases. And those are disorders of the extracellular matrix. These are kind of sickish, overactive fibroblasts that make abnormal and pathogenic matrix. But what we've learned at looking at some of these diseases that this is not an irreversible process. These fibrotic matrices are actually quite active in turning over, and so they're not kind of put there cast in stone and never move. This is still a biological tissue that can be addressed and can be expected to also change for the better. So I think that's another aspect of the disease that potentially could be addressed even later on in the condition. At least that's one of the hopes here. One interesting aspect, of course, where we talked about editing, gene editing and transcripts, and so and AAV gene therapy. When this gets a little bit into kind of gene therapy biology, but when you do an AAV gene therapy and you deliver a DNA to the cell, it gets to the nucleus of the cell, but it doesn't integrate into the chromosomes of the cells. It lives as its own DNA, as its own gene, which is fine as long as the cell does not divide. If the cell divides, you're actually losing it because it doesn't get replicated with the chromosomes. You just kind of dilute it out and you lose it. And that's perfectly fine if you think about a nerve cell that will never ever divide after you are born. Every nerve cell stays the way it is, and if you get in there and treat it, that's good because the cell will not divide. However, in muscle, that's a completely different story, and in fibroblasts too. These are cells that are proliferative, that can expand and divide. And if you have a gene therapy that is not in the chromosome, you can lose it. if you have a gene therapy that edits the chromosome, and that's a pitch for gene editing approaches, is that actually works to your advantage, because now the cells that are corrected, when they proliferate, all of their offspring are corrected. And so you can amplify the genetic effect doing that. So there's pros and cons to both scenarios where the cells never divide and where the cells continue to divide depending on the genetic approach that you take. Any? Shoot me down. No? Okay. Tina.
I'm just going to add a non-medical treatment kind of statement because I can't help myself. So as we're talking about stem cell therapies and muscle differentiation as well as stem cell differentiation, it needs some kind of stimuli, right? And so I think whenever we're looking at medical treatment, we should also look at muscles do not get stronger, fibers do not get bigger without some kind of stimuli. And so that's where, don't forget the rehabilitation part of medical treatment. So combination treatment is not just medicine. Exercise is medicine as well. And so as we talked about earlier in some of the other sessions is how much, and we need to look at it from a dosing perspective because exercise is supposed to decrease inflammation, improve regeneration, decrease oxidative stress, but at the right dose, right? And so some of the things that we are looking at some stem cell therapy as well in a different disease, but it is really, it's that fine tuning between how much. And so I just want to put the pitch out there that as we're looking at this as families, what can you do today is really fully engage in making sure you maintain your musculoskeletal health while these treatments are being investigated.
Yeah, no, this is absolutely right. And it's a treatment in its own right, which has a genetic signature. If you do exercise and if you use your muscle, the genetic signature in the muscle changes. Its energy changes, its mitochondrial content changes. And we said collagen 6 and also collagen 12 are atrophic muscle diseases from the get-go. That is a part of muscle disease that you can much better address with exercise than a dystrophy where degeneration happens. And so absolutely, I think that's here now and should be used now. Thanks for making that comment. So trial readiness, and I saw Reghan Foley walk into the room. Did she disappear again? Where is she? Oh, she just left. Okay. Yeah, exactly. That would be a good time now for her to talk about it. So one thing that gets us clinical trial ready is what Tina and other physical therapists do, which is to develop methods to measure outcomes appropriately. So that is, if you do a therapy, it becomes an experimental therapy. That is, even now physical therapy methods, you want to know whether you're doing something that helps the disease, something that makes no difference, or something that gets worse. And so you need to have appropriate outcome measures to study this. And how do you do that? So while Reghan is coming back, maybe Tina, can you comment how you develop outcome measures and why they are important?
I wasn't prepared for this, but is Reghan back? Okay.
Rachel stole me away, sorry.
So really with outcome measures, it's really harping on some of the most boring things. But if you think about outcome measures, is how reliably can this be done from me, from someone who hasn't done the outcome before. So you want it to be stable. You want to understand that it is measuring what you think it's measuring. And so when you think about outcomes development, what is it that you're measuring? And that comes from the community. You have to tell us what is important, what's important to change, because if we don't hear from you, we're going to measure the wrong thing. And if we measure the wrong thing, how do we know something's improving, right? And so once we do define what the right thing is, then we have to prove that it's measuring what it is. And so that sometimes that takes 10 years. And so this has to happen in parallel to drug development. And so what we do is, like, is it reliable? Is it valid? So measuring what you think it is compared to, is it actually measuring muscle force, or is it measuring something else? And how can we standardize it in a way where everybody's measuring it the same? And then with our learnings from SMA and Duchenne, once a drug is approved, what happens after the regulatory agency said, great, you can have treatment. Now you have the payer sources as well saying you need to do this measure. And if it gets worse, we won't renew your treatment, right? And so there's a lot of impacts on clinical outcomes development that may just seem like, you go to clinic and you're just measuring muscle strength, but if I'm measuring it different from the next clinician, that difference isn't because you changed. That difference is because we're different, right? And so those are the things that, it's the nitty-gritty and the details that is part of clinical trial design, that this, what we call variability or the reliability in a measure, impacts how long a treatment trial is and how many patients that are needed. In diseases that are ultra rare, you want that number to be really tight so your sample size is smaller. And so we can get to the finish line faster with less time and less patients.
Great. Thank you. Yeah, we can wrap up now.
Yeah, so let me wrap up. This is the reason why that is so important also is because if you want to engage any biotech or any even granting agency or anyone to support a therapy, they want to know these numbers. They want to know whether you know your disease well enough and can study it well enough to show that it works or doesn't work. Right? That number needs to be there. And so thank you all for everyone here in the room participates in that project right now to be ready for that. And then the other thing that's usually helpful in developing therapies is a biomarker, so biological readouts of the disease that you can study easily in blood and urine. And so Reghan Foley is working on that. That's work in progress. Probably at the next QCMD conference we can show you those results. And with that, I thank our entire panel and the entire room for this afternoon.